Uh, yeah, great. So, hello, welcome all to this talk organized by WiPMA, that is Women in Physics, Math and Astronomy group at Aiello Mohali and the QTpi group at Aiello Mohali. I am Prajakta. I'm a third year undergraduate student at Aiello Mohali and I'm here from the WiPMA side. And with me is Sonal from QTpi. Hi, Sonal.
Hello.
Yeah, so we will be the hosts for today's session. So for today's talk titled as Quantum Biology, How Nature Might Be Optimized to Harness Quantum Mechanics, we have with us Professor Clarice D. Aiello from University of California, Los Angeles, or UCLA, as many know it. Professor Clarice is a quantum engineer interested in how quantum physics informs biology at nanoscale. She received her PhD from MIT in electrical engineering, and she also held postdoctoral appointments in bioengineering at Stanford and in chemistry at Berkeley. She joined UCLA in 2019, and at UCLA, she is currently leading the quantum biology tech, that is Qubit Lab, and it is our great pleasure to have you with us, Clarice. Before moving to the talk, I would take this opportunity to also shortly introduce the audience to the WiPMA group at Aiello Mohali. WiPMA, short for Women in Physics, Math and Astronomy, is a collaborative initiative between three academic clubs, the Physics, Math and the Astronomy Club at Aiello Mohali. We aim to encourage undergraduate women to continue their careers in physics and math while providing a safe space to talk about their issues that are especially faced by women in these fields. Similarly, I will hand it over to Sonal from QTpi to introduce the QTpi group. Sonal, over to you.
Yeah. So last year, so we're all graduates from last year from Aiello Mohali itself. We're all physics majors, and we're just a bunch of people who initially we started out. We want to help out our juniors figure out how to get opportunities, how to get admits, and even younger students how to figure out what they want to do and where to get their internships, how to get the internships. And then we thought, oh, we'll make these interactive sessions where we'll introduce our research, and gradually when we ran out of, like, all of us had given talks, so like, oh, it'll be nice to introduce the students to research from other people as well. So that's how we started. And yeah, we like kind of try to have a talk every month, and we're all grad students, so it's like harder to keep track of more talks than once a month. But yeah, and I'm just really happy we're having this session. And yeah, without further ado, I would be very happy to give the floor to Clarice.
Cool. Thank you both. It's I'm really happy to be talking to you today, and I hope that by the end of the talk we can get some time to talk about like my very uneven career and how I got to where I am. And yes, I started at UCLA in November of 2019, namely like 3 months before the end of the world, 3 months before COVID. So it was a really hard time to start, especially when you're in the business of building experiments, which I am. So I will start with my spiel here. At about a little bit just before halfway of my talk, I'll stop for questions, and please, I would like to hear more about each one of you and your questions on the talk, and let's start this conversation, right? So yes, I like to call myself a quantum engineer. This means that I build apparatuses to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything big around us. So people get a little bit, surprised when I tell them that I study things that might be happening inside birds and butterflies. So that's a little bit weird. So before I explain where I'm going, I really need to tell you where I come from. So we're going to start talking about hardcore quantum mechanics, and by the end of the talk, we will have talked about things of biological relevance, such as organismal migration, how our cells respond to oxidative stress, to radiation, and I hope I will have convinced you that I think really we can learn with nature on how to build better technologies. And that's because I'm a quantum engineer interested in how quantum physics informs biology at the nanoscale. So I like to kick off things by saying that I think humankind is obsessed with measuring things better because this might mean that we understand things and nature better. We can think, for instance, about measuring better frequencies. What you see there in the picture is a atomic clock that sits at NIST, which is the American's National Institute of Standards and Technologies, and it lets us define very precisely what a second or what a hertz is using atomic transitions. You could think about ways of measuring better magnetic fields so that the image of your baby is better resolved. And yes, this is the magnetic resonance image of a baby inside the mother's belly. Or you could think about maybe finding ways of building better accelerations so that your gaming experience is enhanced. Those tiny little accelerometers are now ubiquitous in all our handheld devices. But the question that I ask is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity that you want to measure is very, very small? I'm going to argue that in this case, you need a very little sensor that can measure very little things. If the sensor is small, let's make it tiny. Let's actually make it quantum. There's actually a whole body of mathematical arguments that can actually prove that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. In my past, I worked with such a technological quantum sensor. I worked with a single electron spin in diamond that could be put to use as a quantum enhanced magnetic field sensor. And I will need to start talking to you about this technological quantum sensor that I work with. So this is the promise, right? That the spin in diamond can be put to use as a quantum enhanced quantum sensor. So spins, as many of you or most of you might know, is a fundamental quantum mechanical property of matter that is as fundamental as charge and mass, right? Spins measure how how well quantum objects interact with magnetic fields. Electrons have spins, atomic nuclei have spins, and all sorts of quantum objects have spin, even if it's a spin of 0. And physicists and chemists and engineers usually depict spin as, say, an arrow, right? And I bring my props, so you should be looking at, I'll be using them more. So you should be looking at like my spin arrow here and like spin up or spin down just represent two different energy levels of this merely quantum mechanical property called spin. So, but again, let me tell you what sort of electronic spin in diamond I played with and how we can put it to use as a quantum enhanced magnetic field sensor. So the spin I worked with arose in the form, arose associated with a crystalline defect in the diamond lattice, right? And solid lattices, crystal lattices have plenty of defects, and a family of such defects are called in diamond, at least, color centers. Okay, color centers are crystalline defects that they're responsible for the colors of those diamonds that you see there at the bottom, and they're called as such. They're called color centers because they have this very particular property that can absorb light, get excited, and then de-excite, emitting this light back, emitting fluorescence. So that's why they're called color center. Okay. The very particular defect that I worked with is a very hyped defect. It's called a nitrogen vacancy center, and it arises naturally, but it can also be engineered. So that's very convenient. And it takes place when a vacancy, that is a missing carbon atom, sits next to a nitrogen, that is the most commonly occurring substitutional impurity in the diamond lattice. When all those two things comes together, there's like a mess of electrons that interact there locally, right? The vacancy have like nearby unpaired electrons. The nitrogen has like one extra electrons that might be nearby charges in the lattice. So it's really like an electronic mess. But as you start calculating the quantum mechanical energy levels of this apparent mess, something almost magical happens. When you start calculating the quantum mechanical energy levels of this atomically sized thing, it turns out that such quantum mechanical energy levels really look like the quantum mechanical energy levels of a single electronic spin. Okay? So it's very convenient then, because you look at this thing where many electrons interact, and actually you can take it as the you can understand this, and you can effectively think of it as a single electron spin sitting in this, like within this mass of electrons there. So you can take this atomic defect and think of it as a single electronic spin. And the way that we find those things in the lab is actually pretty simple and pretty crappy, actually. So everything happens at room temperature, and you take like your diamond slab. It doesn't need to be like any particular kind of special diamond. Nowadays, people are making diamonds with those defects, but many naturally occurring diamonds actually have those defects. And the very first diamond sample that we worked with was like a jeweler's discard or something. So you take your diamond sample, you put it on top of a moving stage, and you move it around at the same time that you apply a laser that you excite this diamond sample in the lab, right? So actually it doesn't need to be a laser, it's just more efficient and more convenient. But the idea is that you're moving your sample at the same time you're shining laser. The moment that you actually hit one of those defects on your sample, you would expect the laser, because the defect is a color center, the laser to be absorbed, right? The defect gets excited and then it emits back fluorescence, and this fluorescence we can actually collect. So this is what this looks like at increasing zooms. To the bottom right there, you see a blob, right? Which is very interesting because that is the fluorescence signature of a single electronic spin. It's the fluorescence signature of one single nitrogen vacancy center in diamond. Now, the defect itself is much smaller than this. It's atomically sized, but what you're seeing there is a diffraction limited blob, right? So this is how you find those defects in the lab, except that it gets way better than this, right? For reasons that I won't have time to explain, this defect has what people in the field call a quantum state dependence fluorescence intensity. This means that just by looking at how strongly that blob is emitting light, how many counts you're receiving in your computer, you can actually infer if the spin was up or down. Just by looking at fluorescence intensities, you can actually read out the state of a single electronic spin, which again is very different, say, if you think about spins in magnetic resonance, where you need gazillions of spins to get you an electronic signal of the statistical state of those spins. Here, you can actually, by looking at fluorescence intensity, have access to the state of one single spin. And this is something that makes those spins in diamonds very nice to work with. But here's the promise that we could put it to use as a quantum enhanced magnetometer, and here's how this works. So there I depict the two spin energy levels that you can think about as up and down in this effective spin. And it turns out that the energy difference is in the microwave regime. It's about 3 gigahertz, everything again at room temperature, right? And it turns out that one of these energy states, the one that I call 0, is insensitive to magnetic fields. This means that in the presence of a magnetic field, if the diamond that contains this effect is immersed in a magnetic field, state 0 will not care. However, state 1 is sensitive to magnetic fields. That is, in the presence of a magnetic field, state 1 there will get promoted, will get shifted by a tiny quantity that I call their delta. And you might have seen this before. This delta via something called the Zeeman interaction is proportional to the magnetic field strength. So that's a point that not a lot of people make, but that's super important, right? This is actually how all quantum sensing works. The problem of, in this case, of measuring a magnetic field is actually mapped onto the problem of being able to measure a detuning, a shift delta from a known energy resonance, from a known energy difference in the absence of a magnetic field. And again, this is how all, I'm not sure if it's all, but like, I bet my research money that 99% of quantum sensing is just like this. You have a resonance to energy levels that are shifted in the presence of the quantity that you want to measure, be it magnetic field, temperature, electric fields. And basically, if you know how to measure this energy shift, you can know how to measure that quantity, but because you know how the energy shifts in the presence of that quantity, right? So quantum sensing is actually if you will, a signal processing problem, right? You know how to measure those magnetic fields, those quantities, because the experimentalists know, and engineers and physicists have known how to measure detunings from a known energy difference from many decades, right? So that's sort of the idea of all quantum sensing. It's really a signal processing problem. But this very nice way of measuring quantities, of quantum sensing quantities, can only be applied for reasons that I won't have time to fully explain either, while the object is well described by the laws of quantum mechanics. Everything that starts quantum dies classical, right? You can only obtain quantum enhanced information while this spin is well described by the laws of quantum mechanics. If you remember from your quantum classes, this means while there is phase coherence, while this spin can be can sustain a superposition between the states 0 and 1. At some point, after it loses its quantum character, it can no longer be in a superposition. It's better described by classical mechanics as a tiny little magnet that can be like up or down, but not in a superposition state. The time that it takes for those quantum objects to lose their quantumness, sometimes referred to as coherence times. And the coherence times for this naturally occurring diamond at room temperature in the bulk is about 2 microseconds. If you know what I'm talking about, this 2 microseconds is about T2 star times. Okay, and like today people are making better materials to extend those coherence times, but 2 microseconds at room temperature is already incredibly long, right? This means that if you have your quantum signal as a function of time, like after, statistically speaking, about 2 microseconds, your signal is 0, so that acquiring your signal for longer, if you Fourier transform, it won't help, right? So everything that you do sort of happens, the nice physics, the nice information that you can get from the sensor only happens before those 2 microseconds. So actually, the whole game then for quantum computing and quantum sensing is learning how to control, how to kick this spin so that it's well described by the laws of quantum mechanics for longer and longer, so that your signal, your quantum signal, takes longer to go to 0, so that the longer you can acquire, when you Fourier transform it, the better your resolution in frequency, right? Because remember, you're trying to measure a frequency shift. And again, people have known how to kick quantum systems so that they keep quantum for longer, for many decades. Actually, since magnetic resonance was invented almost 80 years ago, right? And the way that you can kick quantum objects made out of matter so that they can keep their quantum character for longer is with tailored electromagnetic fields. And conversely, if you have, for instance, if you have a quantum object made out of light, made out of electromagnetic fields, the way that you can control them is with matter, is by putting like slabs of material in their way to control them. So like you control quantum objects of matter with light, and you can control quantum objects made of light with matter, right? But the idea here is that you can give kicks in the same way that you can give kicks to a swing at particular times, so that you can keep the quantum character going for long. And you do this with engineered electromagnetic fields. So this is a field that has been going on for a long time. So I'm going to show you one way of doing those kicks experiments so that you can keep the quantum keep the spin going on as a quantum object for long. So I'm going to go into the most technical slide of my presentation, but don't lose the overview. The overview is just having a tiny sensor that you can bring in proximity to a tiny little sample, and that you can measure magnetic fields caused by like tiny little things in your sample. So this is the big overview. Don't lose it. Now I just want to show you one way to kick those spins to help us measure better magnetic fields. Okay. So in my past, I used a electromagnetic kicking sequence known as the rotary echo. Okay, this pulse sequence is depicted in magnetic resonance jargon to the right. Here's what this means. Okay, this means that I have my diamond, right, that is immersed into a magnetic field that I want to measure, and I am actually applying a constant wave magnetic field, like a constant magnetic field, like microwave. I'm actually applying microwaves onto my diamond, and those microwaves, they are always on, and they're tuned to the unperturbed energy difference between the two spin states. Okay, so the microwave is always on. At particular times, I actually do a phase kick in this microwave. I'm applying a certain microwave, and then at a particular time, I shift the envelope. I do a phase kick, and then at particular times, I do another phase kick up, back and forth. This sequence is known as a rotary echo, and it has been used in nuclear magnetic resonance, like in the past, to correct for driving field imperfections, to correct for imperfections in your microwave source. Okay. It turns out that this sequence is also known not to correct for detunings from your resonance. And if you remember the tunings from your resonance, those deltas is exactly what we want to measure. So let's see if we can apply this type of electromagnetic kicks and how we can obtain magnetic field information from that. So this is the signal that you obtain from your spin, and the signal is just normalized fluorescence. Okay, if you remember, this spin has a different fluorescence intensity if it's in spin up or if it's spin down. So this is just like normalized fluorescence as a function of how long I actually apply this electromagnetic kicks while the diamond is immersed into a magnetic field. Okay, so this means that when the signal is, say, at the top here, up, the spin is, say, in one state. If the spin is like, if your signal is, your fluorescence is at the bottom, the spin is down, and every mean fluorescence intensity in between signals that the spin is in a coherent superposition of zero, like up and down. For those of you who remember what this is just a Rabi experiment that has some modulations to it. But the thing is that we can actually do a mathematical formalism called average Hamiltonian theory, so that you can actually infer how the signal depends on this tiny little delta, this detuning that we want to measure. Okay, it's a complicated formula, it doesn't matter, but basically this delta only gives you a beat in your signal, but we can now know how the signal depends on the thing that we want to measure. So actually, we can actually Fourier transform the signal, get something that looks like a spectrum. It doesn't matter like the details, it's basically a spectrum, and you can find different peaks in the spectrum. You know well, I know I'm not telling you how, one can know how to read those peaks so that you can actually get frequency information that is then mapped onto magnetic field information. For example, with this spectrum there, you are measuring 3 different magnetic fields, one of which is about 1/10 of the magnetic field of the Earth, which is very, very tiny. But again, don't forget the big picture. Big picture is having a tiny little sensor that you want to kick and keep it as a quantum object for longer, so that you can measure for longer and longer magnetic field information coming from tiny little samples, right? Again, I just want to open for questions now, but I just want to make sure that you agree with me that I've just presented you with a quantum sensor that works at room temperature and under noisy environments, in bulk, right? Close to a noisy sample, and that's very important. So before I open up for questions, I want somebody to start thinking about it. Well, actually, I want everyone to Google this information and to think whether this is a paradox or not. I've just described you a quantum sensor, right? That has a frequency difference of about 3 gigahertz, and it's immersed. It works at room temperature. So, can someone Google what thermal energies are in gigahertz units, like kBT in gigahertz units at room temperature? Okay, how does that compare to the 3 gigahertz of the frequency difference of my quantum sensor? And is this a paradox? Does this bother you? Okay, so if you don't have questions, you can start thinking about those what I just asked right now. But otherwise, this is a great time for me to pause for questions. And I see one question in the chat, so feel free to pause me and just shout out your questions or put the questions in the chat. The first question is, what does the red and the blue curve signify? The blue curve is raw data, and the red curve is the fitting from this formula. From this formula, this is technical, which is a fitting for first order approximation, first order in delta divided by the Rabi frequency. So it's an approximation. What are your other questions? And if you don't have questions, I want to hear your opinions about this pseudo paradox that I just asked you.
Clarice, there is one more question in the chat.
Are there any quantum sensors to detect electric fields too? Yes. And actually, for example, the nitrogen vacancy center can now be put to use as a quantum enhanced electric field sensor. And again, the basis of why those things work is the exact same. Energy differences that shift in a predictable way with the quantity that you want to measure.
Hello. Am I audible?
Yeah, I'm good.
Yeah.
Yes.
Hi. Yes. So I have a question. Like, you say that this sensor is able to detect the change in magnetic field due to the sample. But like, how do we know for sure that it is because of the sample? Like, are we coupling the sensor to the sample by like some process or something?
Okay. So several layers to the answer. First of all, what you can do is characterize your sensor far away from the sample, right, that you want to measure. And you're right. Every sensor inside the diamond lattice might be slightly different because it sees slightly different magnetic field environments. For example, in naturally occurring samples, this spinning diamond, this electronic spin can couple to nearby nuclear spins and so forth. And those things, yes, give you like a residual background field that is always there, right? And that's putatively different for each sensor in a sample because every solid state sample is different from another. Something that, apart from doing sensor characterization before you bring it close to your sample, what people are doing now are building diamond materials that have very, very few magnetic impurities, so that you can bring this background down to very, very low levels as compared to naturally occurring diamonds. So that's one part of the answer. The second part is that, yes, what people do is basically bring this diamond in very close proximity to your sample. There are AFM tips right now that have a nanodiamond containing one of those defects. There are people shooting nanodiamonds containing one of those defects inside cells. So yes, basically you do bring those sensors in close proximity to the sample. And actually, it wouldn't work if those things weren't very close to the sensor. This sensor is very good for a proximity stuff. This is a proximity sensor if you want.
Okay, thank you.
And there's another question. Matthew asks, do you think we're better at optical measurements than we're trying to convert the domain of measurement while measuring the magnetic field with the sensor? I am not sure exactly if I get the whole question. I think the question is, please, Matthew, correct me if I'm wrong. I think the question is it easy to do optical measurements than electrical measurements on the state of a spin as one would do in nuclear magnetic resonance, for example? If this is the question, the answer is usually yes. So the technology to detect photons and single photons is very advanced nowadays, and it's probably easier it's more sensitive than the technology used to detect spins via precession of magnetization as is used in NMR for single spins, right? So I think technologically we're much more advanced to do optical measurements. It's much more convenient, right? And this is one reason why doing optical measurements for spins is so cool. Let me know if that's not the question, because then the answer will be different. Okay, that was the question. Awesome. Okay. Sukesh has a question. Can any material be used? Because Zeeman effect can be observed only in certain materials, right? So the thing is, like, any magnetic field is going to couple to your electronic spin in a way that is described by the Zeeman interaction, right? So regardless of what causes this magnetic field, your magnetic field is going to be coupled to your electronic spin, which is the quantum sensor. And then you can always get information on that magnetic field using your spin quantum sensor. So basically all you need is a magnetic field source. I'm not sure if that answers, but let me know if it doesn't. So anyone has an idea of, please put in the chat if you Google this information. What are thermal energies kBT at room temperature in gigahertz units? Can someone enlighten me and put it in the chat, or unmute yourselves?
It's about 6,000 gigahertz, right?
It's about 6,000 gigahertz. So let me tell you, let's recap, right? So we have this 3 gigahertz quantum object immersed at room temperature in a 6,000 gigahertz thermal bath. Am I saying that we're going to get useful information from this beast? What about thermalization? Isn't this thing going to thermalize? Yes? No? Is this a paradox?
It will thermalize, right?
Yes, it will thermalize. So does this mean that it's useless as a quantum sensor?
But like
Yes.
it will thermalize at long times, right? Like so until it does, we can use it as a sensor, I would say.
Yes, absolutely, right? This is actually a one question that I usually get from people, but how on earth, right? How on earth are you telling me that this tiny little sensor immersed in a much hotter bath, how on earth does this thing work as a quantum sensor? How does quantum mechanics work? And Anubhav said it all.
Yeah, Anubhav. Yeah.
Anubhav said it all. Like, I can only obtain quantum information out of this object before it thermalizes. After it thermalizes, it stops being well described by the laws of quantum mechanics. The important part is, like, in those 2 microseconds that I have to play with, that's a lot of time for me to play with and get quantum enhanced information. After thermalization, all quantum advantage is absolutely lost. So there is no paradox here. There are quantum objects that are immersed in much hotter bath environments that can provide quantum information for those short times. So yes, Sahil says the sensor is only active for 2 microseconds. Yes, the idea is that you can only obtain quantum enhanced quantum information for those 2 microseconds and not longer. Okay, and that's important because those 2 microseconds matter. Okay, so this is not a paradox. More questions, or can I?
I think we can go on to talk.
Okay, so now, oh yes, question.
Yeah, I had not a question, like just a
Yes.
comment or a query. Like, so when the sensor thermalizes, can we like somehow not bring it back to the like non-thermal state? Like
we can.
if we measure it or something. Exactly.
Yes, and that's exactly what we do actually. When it thermalizes, we go back into the cycle and take another measurement and so forth. That's exactly how it happens. We can cycle it back.
Perfect.
Right. But we're only relying at those 2 microseconds, roughly 2 microseconds per experiment run, right? And no longer than that. Sarbojoy, am I pronouncing it correctly?
Yes.
Tell me. Question.
Clarice, my name is Sarbojoy Das. Am I audible? Am I audible?
Yeah, you're audible, Sarbojoy.
Yeah. So what I wanted to ask was, like, we do experiments in spectroscopy where we have coupling between, say, 2 different states, right?
Yes.
Like, suppose you have one magnet, like one such magnet.
I'm just trying to put my speakers close. Sorry. Yes.
Yeah. Suppose you have this one, like you're trying to detect one of the magnets, right? In a sense, right? Now you have others around, and suppose if I wanted to detect any coupling between them, what would be the time to detect them? I mean, once the system thermalizes, you have semi-classical, classical things coming in to play. But suppose if I had to detect a coupling between, say, say between R1 and R2, in a sense, if there were in some sense, this is like, in some sense, this is like pump and probe in spectroscopy, right? Just different from optical, like this is in the optical regime.
Well, that's I'm not sure. I will think about this. I had never thought about this. I do not know if it's like pump probe spectroscopy. I mean, it's not like pump probe spectroscopy in that you're depleting one and measuring. It's not like this, right? What you're measuring is how your sensor.
So I'm just asking about if there is any, if I had to detect, suppose in the diamond, if these magnetic centers are there, right? And there might be coupling between one or more magnetic centers. So if I had to detect any
one or more
signature
sensors
of these, I mean these, I mean when you have the sample, that is what you're doing, right? Like you're trying to.
So first of all, usually, so there are 2 modalities. Like you can usually, if you want to sense using a single electronic spin, what you want is to either find one that is isolated from other nitrogen vacancy centers, or now people are like they get the diamond sample and they actually make defects at particular places, and they're far enough so that they do not talk to each other. If you're using a single nitrogen vacancy center to measure things, it's best if they do not, like, if they're not in close proximity to another. There is another way of making measurements that I'm not discussing here, which is by actually making a sample very, very, very packed with many, many of those nitrogen vacancy centers. And in fact, you use like an average of sensed magnetic fields over those many spins to get like a result. So basically, I'm not sure I understand, because you're concerned about the coupling among different
It's okay.
nitrogen vacancy
I'll
centers.
ask the question later, maybe. No,
Cool. Okay.
the question was the same thing. Like, so you were talking about like putting them far away. So these nitrogen centers are, far away from, so that they can't so they can't talk to each other in a sense. But what if they were to talk to each other, and then I would have to detect any the signature of the coupling between them? What would I need to do in that case? That is what I was asking.
Sure. Well, in this sense, if you have a lot of nitrogen vacancy centers together, what you're reading is sort of a statistical thing of like how each individual NV center responds to both the couplings to their environments and to like the magnetic field that you want to measure. Right? I think that
Yes.
the idea here is like the differences in order of magnitude of, like, if you will, the background and the thing you want to censor. But we can talk about that more. And there, yeah, I'll defer that a little bit, if that's okay. Cool. So now I get to the part of my talk where I really want to talk about, right? So up to, like, I don't know, 5 years in my life, those, like the sensor that I just described, these sensors that you see there, those were the type of sensors that I was used to dealing with. But at some point I realized that sensors made by nature many times outperformed the humankind made sensors in crazy ways, right? So I work with a nature-made magnetic field sensor that I hope I'll convince you that it's actually, it works very similar to the nitrogen vacancy center in diamond. Okay, so I think that this is like really the official start of my talk. So, but in order to talk to you about this nature-made magnetic field sensor, I really need to start talking about biology at the nanoscale, namely the chemistry. So it's actually known in the field of basic chemistry for many decades now that magnetic fields can alter the final products of a class of chemical reactions that are sensitive to spin. Okay? I'm giving you the summary of this, but if you're curious about that, I can do a proper like demo with plenty of spins later on. But the idea is that there's a class of chemical reactions that depend on spin in this particular way. There's a chemical reaction happening, and at some point the chemical reaction comes to a crossroads. And at that crossroads, if a certain electronic spin, and I'm going to use jargon, is measured by the environment to be up, the chemical reaction continues through one branch. If at that crossroads, the spin state of a certain electronic spin is measured by the environment to be down, the chemical reaction continues through another branch. Importantly, the final products of those two branches are macroscopically different. And external magnetic fields acting at that crossroads short time, the time of the decision, right? A tiny magnetic field can actually influence if the chemical reaction is going to go from here or here. So an interaction with a magnetic field for very short time can have macroscopic consequences because it changes the macroscopic final products of a chemical reaction, and at much longer downtimes, like times, right, downstream, because it happens downstream. So this has been known again for decades now. This has been demonstrated at room temperature, in solution, in the gas phase, in the solid phase, and for magnetic field strengths down to the strength of the magnetic field of the Earth. Okay, and for reasons that I won't have time to explain, but again, if we have time, I can do the full explanation. This is eminently a weak field phenomenon. Okay, this means that this type of chemical reactions that are spin sensitive are only active for magnetic field strengths between microtesla and millitesla. Okay, the magnetic field of the Earth, for comparison, is about 50 microtesla. So this means that if you put those chemical reactions in a test tube inside a magnetic resonance 5 Tesla machine, changes in magnetic fields will not alter the fate of the chemical reactions. However, weak fields on the order of those produced by those things around us can actually shift the balance of those chemical reactions. Again, this has been demonstrated. Okay? But this is where the biology comes in. And again, in all truthfulness, sorry, I really don't care about birds, but birds is where this whole shebang started. Okay, so it's been known again for many, many decades that birds, when they migrate, they follow, at least as a partial cue, the magnetic field of the Earth, which again is weaker than the magnetic field that you sense when you put your cell phone close to your face, which is crazy to think that birds do this, right? So how might they be doing this? There have been many explanations that have been sort of shut down by experiments, but in the end of the 70s, some brave theoretical biophysicists, at that time, people were like, oh, those people are crazy. They made this outrageous hypothesis. They hypothesized, well, were the same type of spin-dependent chemical reactions at play under physiological conditions inside the freaking birds, birds and organisms in general could sense magnetic fields to the extent that it could sense different physiological concentrations of products coming from these spin-dependent chemical reactions. So something that was hypothesized, it's not proven yet for birds, but for example, should some sort of chemical modulate the photo response of birds in a magnetic field dependent way, birds, when they shift their heads right and left, if there's a chemical that modulates how sensitive the bird's vision is to light intensity, maybe the birds could see magnetic field lines due to those modulations in light intensity, right? This has not been confirmed nor refuted yet, but people like experimental biophysicists started looking for proteins that could sustain these spin-dependent chemical reactions. And at that point, the only animal, because birds, the only animal protein that could sustain those spin-dependent chemical reactions was a protein called cryptochrome, which is a fluorescent protein. Okay, that means it absorbs light, gets excited, and then fluoresces back. It does this because it has a flavin, that's a pigment. Okay, and this protein, this only one that at that time was known to be able to sustain those spin-dependent chemical reactions, was called cryptochrome, and it's present in the bird's eye, in the antennas of migrating butterflies, in our eyes, and it's actually present in all our cells in our body because it's responsible also for circadian rhythm regulation. Okay, and actually it turns out that cryptochrome is very widespread throughout the tree of life. All those organisms there have expressed cryptochrome. Okay, and it turns out that there are many experiments done in organisms that express cryptochrome that ties the presence of the functioning of cryptochrome in how those organisms interact with magnetic fields. All those organisms boxed in red have to do with organisms that seem to be sensing magnetic fields somehow in conjunction with cryptochrome. And yes, humans are right there too. And in fact, there's a lot of evidence for cryptochrome-based magnetosensing, but unfortunately, at very disconnected length scales. Like there's evidence at the chemical level, and there's evidence at like the whole organism level. So let me talk to you about the evidence at the chemical level for like cryptochromes in solution. And there's actually no doubt that cryptochromes in solution behave pretty much as the nitrogen vacancy center in diamond was behaving. Okay, for example, if you look at the top left picture, what you see there is an experimental result for cryptochromes in a vial, where where experimenters shown shown a laser onto this cryptochrome so that it could get excited, right? Absorb this light, it's a fluorescent protein, absorb this light, get excited, and emit light back. So what you see there is the fluorescence of cryptochrome in solution as a function of the time during which you are shining the laser. The first thing that you see is that this fluorescence is going down. You're killing the pigment. It's called bleaching. Okay, that's not important. The important part is what happens with this fluorescence as the experimenters start pulsing a tiny magnetic field on and off close to this vial. What they see is that this fluorescence actually gets modulated, as you can see in the inset, on and off, and it actually follows the magnetic field, the magnetic field shape. Okay, this is not an artifact. This has been measured with different magnetic field intensities, frequencies, and this, I want to point out, is in direct analogy with the freaking nitrogen vacancy center in diamond. Just by looking at how much the nitrogen vacancy center was emitting light in the presence of a certain magnetic field, you could infer whether the certain spin was up or down. Here, just by looking at how much the freaking cryptochrome solution is emitting light, you can actually infer if the spin was measured by the environment to be up so that the chemical reaction went one branch, or if the spin in question was measured to the environment to be down so that the chemical reaction continued through another branch. Okay, so this means that by looking at fluorescence intensities, you have quantum information out of this, the chemical reaction sustained by this cryptochrome. Also, there's not complete evidence, but decent evidence saying that cryptochrome in solution might be coherent, might have coherent times, might be quantum for about 1 microsecond, again, at room temperature, which is on par with the 2 microseconds that I quoted for the crappy nitrogen vacancy center in naturally occurring diamond. So this is super cool. There's no doubt whatsoever that cryptochromes in solution are bona fide quantum sensors. Okay. However, the next step in the evidence that exists for cryptochrome-based magnetosensing only happens at like the length scales of like freaking whole birds, right? So those are very beautiful, very hard experiments. Like people every year during migration season take like n equals to 15 birds, put them into cages, mess up with magnetic fields around, and see which direction the bird wants to migrate out of, right? And the birds sort of respond to the magnetic fields in a way that is consistent with like a quantum process being sustained by cryptochrome. Like flies do not migrate, but flies can be trained. I have no idea how this is harder than like quantum field theory for me. Flies can be trained to find food based on the presence of a magnetic field. Okay, they can be trained to do that. So they're trained to find food based on if there's a magnetic field around. Then the researchers remove the cryptochrome gene from those flies, and the flies can no longer find food in the presence of a magnetic field. Then in a further experiment, researchers put back human cryptochrome inside the freaking flies, and the flies are back to finding food in the presence of a magnetic field. All of this, or most of this evidence, this macroscopic evidence, is consistent with like a quantum sensing like behavior happening at chemical landscapes, but there's freaking nothing in between. And here's where we enter, right? We want in our lab to sort of bridge those two landscapes. We want to be able to do spin experiments inside much smaller living things, right? So basically, what we're building in our lab, we're super behind schedule because pandemic and all, but that's fine. I'm zen about this at this point. We are building glorified microscopes with coils, okay, so that we can at the same time look at spin information via fluorescence and what happens, say, downstream chemically in the cell using regular fluorescence markers, say, in single cells, right? So what we want is to correlate two regimes of fluorescence time scales and intensities. Spin physics happens very, very fast, from nanoseconds to microseconds. And we're talking here about collecting single photons, right? We want to correlate that in the same microscope as we correlate like fluorescence markers of chemical processes inside cells, so that we want at the same time to be able to prepare, manipulate spins, measure that we're doing something deterministic with the spins in cryptochrome, right? And see how that affects other cellular processes downstream. So what we want is hijack instrumentation that people have been using to study and control technological quantum sensors and start studying and controlling those living, those natural made quantum sensors, and start correlating them in the same experiment about what happens downstream, so that we can actually have everyone talk, the chemists and the biologists. So this spin flavor of quantum biology is really right now bottlenecked, right? So most experiments done in this space so far have been done by chemists or biologists. Again, those are different experiments. We think that as quantum physicists, we have something to add in providing high-tech instrumentation to deal with those biological spins in vivo. So when I say I'm going to build microscopes, it's not going to look like a mic. Those pictures are not from our lab either, right? When I say that we're going to build microscopes, it's not going to look like the microscopes to the left. It's going to look more like the thing to the right, which is basically what an atomic physics experiment looks like. So it's an atomic physics experiment, but working on a biological sample. For physicists, I would describe this as controlling light matter interactions where matter is actually biomatter, right? And what's different from usual, again, it's not better or worse, it's just different from usual biophysics experiments, is that the mathematical models actually drive the experiments, right? So in some biophysics experiments, you do the experiments and then you model. Here, we're trying to do something different. Actually, you can model those spin processes. For example, in cryptochrome, you can do open quantum system simulations with a minimum of 3 spins. I won't have time to talk about this, but you can actually build, basically, it's you can solve numerically Schrodinger's equation in the presence of noise. It's like a Liouvillean equation using some parameters of cryptochrome that comes from the published photophysics of the thing, so that you can actually start making predictions. I won't have time to explain a lot, but I want you to look at the top right plot here. This is a prediction that our crappy models have made. We're not the first to make those predictions, right? But I'm dying to take those curves experimentally. So here, what you see in the top right plot is the magnetosensitivity of cryptochrome, like how sensitive the spins in cryptochromes are as a function of the magnetic field strength for cryptochrome, like tumbling in solution. Okay, what you see first of all as a prediction is that this magnetosensitivity is not monotonic; it goes up and down. At about 10 times the magnetic field of the Earth, like at about 500 microtesla, the magnetosensitivity went down. This can be explained by spin dynamics. I won't have time, but in any case, this is what I told you, that those spin-dependent magnetic field effects happen at small magnetic fields. Okay, so this is one thing. The other very cool thing about this curve is that it peaks very close to the magnetic field of the Earth, which is either a coincidence or it actually means that the magnetic field of the Earth might have had some evolutionary pressure on how this thing evolved. We cannot tell like if it's a coincidence or not, but we think that people who do directed evolution of proteins can help us finding those things out, right? But again, this talk is not about birds, and actually it would be a much poorer talk if it were only about birds. The cool thing is that there's again macroscopic evidence that is consistent with spin physics driving many super relevant biological phenomena. Okay, experiments done with, say, plates of thousands of cells or whole organisms seem to be consistent with the fact that things like production of reactive oxygen species, cellular glycolysis rates, cellular acidification rates, respiration rates, stem cell mediated growth in planaria, yield of DNA repair, all those things being sensitive to magnetic fields in a way that is consistent with being dependent on spin-dependent chemical reactions happening under physiological conditions, right? Cellular autofluorescence seem to be magnetic field sensitive, probably in flavoproteins. Cryptochrome is also a flavin, but there are many other flavoproteins, probably like this in this paper that I put there for fluorescence of flavins inside mitochondria, right? And I want to spend some time because people get crazy about this picture, looking at this picture to the bottom there. So that's the story. I have a friend who's a bona fide precision measurement guy. His name is Peter Fierninger, and what he does for a living is to build very good Faraday cages, hypomagnetic chambers. And then what he does is he shops ultra cold atoms experiments inside those hypomagnetic chambers. His hypomagnetic chambers have like noise levels on the order of single nanotesla, right? Again, magnetic field of the Earth is about 50 microtesla. So what he has inside is like crazily tiny magnetic fields, even compared to the magnetic field of the Earth, which is tinier than the magnetic field of your cell phone. So he started hearing things about, magnetic fields in biology. And what he did was grow tadpoles for 2 days inside those chambers. And 2 days was the longest that he could do without needing to ask for a biopermit or something, under 2 conditions. In one condition, he applied inside those chambers a mimicked magnetic field on the order of the magnetic field of the Earth. And on the second condition, he just had the nanotesla field. The tadpoles grown under regular, like mimicked magnetic field of the Earth conditions, they were macroscopically fine, but the tadpoles grown under nanotesla conditions, as you can see in the picture there, macroscopically, about 30% of them were macroscopically deformed. Okay. Now, this experiment does not probe spin physics, but given the fact that you didn't need to give a field, only take out a tiny little field, right? Given the magnitude of the fields involved, everyone's best guess is that this somehow involves some sort of spin physics. My best guess is, so there's lots of spin physics in melanin, the thing that gives the tadpoles, I think, its color. So my best guess is that this involves spin physics in melanin. But experiments such as those, and there are other experiments done under hypomagnetic field conditions that show that epigenetics change, like things called DNA methylation changes just by removing magnetic fields. It gives rise to all sorts of like, it opens up like a Pandora box, right? From like things that we can manipulate here on Earth to like, what's the magnetic field on Mars? Can we colonize Mars? Like, can we grow lettuce in Mars? Can we reproduce in Mars if Mars has a different magnetic field of Earth? And it does. I think it's much weaker than the Earth, right? So there are all sorts of very cool spin physics phenomena in biology that I think should be pursued in a very systematic way. And this is exactly what we're trying to do in our lab in a very high-tech way. Can we establish or refute down to a certain noise level that spin physics accounts for big stuff in biology, right? And importantly, can we manipulate it, right, to technological and therapeutic advantage? So if nature really is using quantum mechanics to function, it knows darn well how to deal with noise, right? And noise is, the quantumness quenching noise is the reason why we don't have quantum computers right now, right? That work all the time. So if I'm not saying like, let's replace Google's beautiful quantum computer with a molecule, but rather, are there strategies being used by nature on how to deal with noise that we could apply on top of existing quantum technologies, right? And also, I think that this is a very promising stuff, right? Can we tune those endogenous quantum knobs in biology to like therapeutic advantage? Again, I don't think this is going to happen in 5 years, in 10 years, but maybe in 50 years, if we start doing research now, right? Can we have like a book in that, well, if you want to drive a certain chemical reaction around, this is the magnetic field strength, frequency, and direction that you need to apply, right? So that we can start applying with like devices that produce, devices that produce like weak magnetic fields, right? Can we locally start driving physiology around? Again, this is science fiction, not going to happen soon, but we need to start thinking about those things now. I think it's possible, right? In half a century or so. And we're actually not the only people starting to think about biology in high-tech quantum ways. There are many very recent experiments, 17, 19, that actually are using those endogenous quantum knobs in biology, right? Some people get super excited when I say, well, that's like quantum optogenetics, you know, like in optogenetics, you have to code in proteins that respond to light. Well, in this sort of modality here, the knobs, the quantum knobs that you want to tune, they are already there. So what can we do with those things? So this is all I wanted to share with you today. I'm sorry I went a little bit over time. I want to thank my group who were very brave to join a group, a starting group in the middle of a pandemic and working on a field that is still very contentious and very misunderstood, right? It's sort of a pity because like if I Google like quantum biology in my browser, like hit number 13 is like some new agey quantum healing. So that's still, it's one of my hats is trying to educate people what we mean by quantum biology. And we mean doing experiments in a high-tech way as possible to really establish or refute how nature is, to which extent nature is using quantum knobs to function. And I like to finish my talks by saying, may the quantum be with you. So what are your questions? And I'll stop sharing so that I can see you.
Well, I am very intrigued. I've like read articles about quantum biology in general. I have seen overviews, but this has been very relatable, especially like because I've had a physics background, and I'm sure people agree.
And it's very hard because I'm sorry, I'm a physicist too, and physicists are the worst. Sometimes they don't even want to engage, right? They're like, oh, quantum mechanics at room temperature. Oh, you're crazy. You're a new agey person. You don't know what you're talking about, right? And the biologists have been doing things chemically for like 50 years, right? You can take a medicine and shift your chemical reactions around. It's very hard to talk to them too. So it's like really no one's land right now. It's sort of frustrating, and it does need there to be like more community building so that people can start talking through those things together.
Yeah, I think it's really important to build the bridges. I mean, that's why interdisciplinary research is so important. I mean,
I know
we can
right?
really help the biologists, and the biologists can help us. I mean, honestly, this can actually be the answer to building scalable quantum computers. Who knows?
I know, and like I don't know how you all think about it, but
Oh, I can't hear you. I'm sorry.
can you hear me now?
Oh yeah, I can.
Okay.
Sorry.
So quantum interdisciplinary research is very, it's a very hard place to be, in the sense that many funding agencies, they do not operate at the intersection, right? So
Yeah.
for some funding agencies in the US, if I apply with a proposal, an interdisciplinary proposal, it has to go through two judging boards, right? And then it's like the laws of like multiplying small probabilities, right? If in any single panel I have like 20% of probability of being accepted, I have like 20% times 20% of being accepted by both panels. So it's a mess.
Okay.
Like quantum biology right now is a very aging field, that people pivot to doing quantum biology like experiments like at the second half, the last third of their career, when they're established, when they have a continuous source of funding. So there's very little incentive for young people, for early career people to jump in, to bring like new technologies, to bring refreshing, right? So last year, we just started here at UCLA, the Quantum Biology Center, which is the first center in the US with goals to improve community building in quantum biology, right? In the sense that what we want to do is like bring people together. We want to like 20 years ago, there were no bioinformatics departments. So what should we start doing now if we want to, in 20 years, to have a quantum biology department, a quantum biology workforce, right, a quantum biology major? So that's the other hat that I wear, telling the public what quantum biology is, what it's not, and how do we start thinking about it in a responsible way.
Yeah, and not as something that's a sub part of either biology or physics or quantum physics. It's a whole new field in itself.
Yeah, exactly. Right. And there's another question. Intelligence at a physiological genetic level has a quantum description. I have no idea. Would it also offer insights into the value of quantum perceptron, the fundamental unit of a quantum neural network? I do not know. I do not think there are any experiments that are thinking about probing this now. And I think that it all goes back to that fundamental dichotomy, right? Even for spin quantum biology, there are experiments in test tubes and experiment in birds. And I think that birds are really huge units for us to think of. Like thinking about a brain is a very huge unit. There's so much more that needs to be thought about at like tiny units of life, say a single cell, 2 cells, a little embryo, right, before we can start thinking about more macroscopic things. And this step is important, right? Because right now there is no unambiguous proof that, say, cryptochrome is working as a quantum sensor inside a cell, for example, right? The evidence is consistent with that, but nobody has measured the spins inside cryptochrome inside a cell as we want to measure, and that's necessary because
yes. So this was like my question that I was going to ask, like, how do we know for sure that it's like cryptochrome is the reason behind we are seeing this? Because biological systems are like really complex. You never know like what is responsible for what exactly.
Never, I'm not sure. Currently we don't. And first of all, I mentioned cryptochrome, which historically is the protein because of birds that people have been studying. But this type of spin-dependent chemical reaction seems not to be confined only to cryptochromes. There are many other proteins, other cellular processes that seem to be magnetic field sensitive in a spin consistent way that do not have cryptochrome. So this is broader than cryptochrome. And again, that's exactly the point, right? How do we prove that it's cryptochrome? Our approach is to like learn how, if cryptochrome is quantum inside a cell, which it may not be, right? We might show that by doing, say, Rabi-like experiments with cryptochrome inside the cell. But what we want is the ability to manipulate cryptochrome in the same way that you would manipulate technological quantum sensors, that is with tailored electromagnetic fields, and see how a certain preparation of spins can actually influence downstream processes that people have in the past thought is involved with, say, the functioning of cryptochrome, right? I don't think this will be like an unambiguous proof, because yes, I don't think we're going to be able to do exactly like the whole pathway, but nobody has shown that spin manipulation, whether or the extent to which spin manipulation changes something else that we can measure in cells. So what we need to do is do a quantum measurement at the same time in the same experiment that like a downstream physiological measurement. And I don't think that's the final answer exactly because of what you said, right? But not even this correlation has been shown. Yes, questions, more questions.
There's another question in the chat. So Pranav is asking if you could give more examples of where we can apply these biological phenomena in the modern day biotechnologies.
Yes, for example, many. Cool. I'll talk about my other favorite thing. First of all, for quantum sensing, right? I showed you that the theory predicts that cryptochrome magnetosensitivity is sort of centered around a certain magnetic field strength. So basically, cryptochrome is a sensor that is very sensitive to a certain bandwidth of magnetic field strength. So maybe we could learn how to make those biological sensors that are sensitive to bandwidths of strength, of frequency, and things like this. Right? The other thing, and I'll talk about something slightly different. There's this other spin-dependent phenomenon that happens in biology that was observed for the first time in 2011, and I think it's going to be the next big thing. It's called chiral-induced spin selectivity. Okay, here's the observation. It was first observed in DNA. Now people have started seeing this in peptides, and now people are actually making nanomaterials that exacerbate the phenomena. Here's the observation. The observation is that if you push electrons through a molecule of DNA, and if you spin analyze the electrons that are being spit out by the molecule on the other end, at room temperature, you would expect, because of thermal energies, electron spin up and electron spin down, thermal energies being very close, you would expect to find more or less 50% spin down and 50% spin up of the electrons that you push through. Except that that's not what's being found. In DNA, people found like a slight polarization. I think it's like 45, 55%. But depending on the material, you're seeing crazy spin polarizations at room temperature of like 90% up, 10% down, or like 95.5. And I like to call it like nature's own way of doing spin initialization, right? It turns out that spins, there is one evidence that spins inside, electron spins being transported inside live bacteria, that they are spin polarized. Okay. So why this happens, I don't want to talk why this happens, because it's not super understood yet. But when people try to put Hamiltonians on why this chiral-induced spin selectivity happens, people end up with Hamiltonians that look like Hamiltonians that are used to describe topological insulators. But I'm not going to talk anything else about that. It is sort of really crazy. And like, can we design materials, don't need to be biological, but that exacerbate this chiral-induced spin selectivity to make qubits talk at long distances, for example, right? If you, back to our question, if you have two electron spins in the envy center that you want to make one talk to each other, if you want them to talk, usually you rely on dipole-dipole interactions, right? Which means that they have to be close by. Imagine that you could have like two nitrogen vacancy centers far away, but separated by a chiral material, right? Like, can we make chiral interconnects that help like information transfer along longer distances? So I think I'm not giving you like the full picture, sorry, but chiral-induced spin selectivity, I think, is going to be something that we're learning with nature that might influence quantum technologies in the near future.
Okay, that was very interesting.
It's
Okay.
crazy. It's crazy. Yes.
Yeah. So there's another question.
Yes.
So Kamish is asking, I think he wants a little more discussion on the epigenetic changes that you talked about in the controlled magnetic field environments.
Yes. So I'll put, let me put there, I put there one review for chiral-induced spin selectivity, and let me put one research done by the Army in 2016. That talks about such epigenetic changes. Again, they do not prove that it's spin, because nobody can prove that it's related to spin, but what they see is consistent with what one would expect. Copy. Yeah. Yeah, it needs a VPN. So those are some, let me, there's more. There's more things related to epigenetics, stem cell. Let me get my, and electromagnetic fields mediate. Okay, those are the titles of the articles. This is from a proposal that I wrote. Just Google all those articles. You see, like, low magnetic field exposure affects the robustness of epigenetic programming and whatnot. Again, none of those articles talk about spin, but the important thing is here is that the magnetic fields involved are very tiny, right? And when the magnetic fields are very tiny, like this tiny, my best guess is that right now is that it involves spin. Again, up for grabs, right? Here's what we're trying to prove or refute in our lab.
Right. Also, that actually relates to another question. Sahil asked if, instead of, because it's not been conclusively proven that it's indeed spins that lead to, that interact with the magnetic field and lead to the different phenomena that we see. So as an analogy, say like how in superconducting qubits, two-level systems interact not via spin. So like there could be other explanations to what the magnetic field is indeed influencing the system.
Sure. For example, for birds, there have been other explanations. So there are three ways. Let's talk about birds. There's three ways that organisms are known to interact with magnetic fields. One is the way that sharks and rays do. They have like a conduction channel, basically, and as they cross magnetic field lines, because of the Lorentz force, there's like an induced voltage, and they can read this voltage. That's one way. Birds are not known to have those channels. They have, if those exist, those have never been found. There's a class of bacteria called magnetostatic bacteria. What they have is like huge chunks of magnetite, of little magnets inside them, organized in a linear fashion, okay, called magnetosomes. What happens is that those bacteria, the chunks of magnetite, they actually align like a classical compass with the field. So that the idea is that the whole thing aligns with the field, possibly presses onto some mechanostatic ion channel. That's not super clear, but there's like a movement of an alignment of big chunks of magnetite inside those magnetostatic bacteria. For that to work with birds would need to have big chunks of magnetite, because otherwise thermal fluctuations would not give like this, the shift, this movement, this net movement when the magnetic field is present. And for migratory species, it's not, such big chunks of magnetite have never been found. Okay. And again, people have put birds to fly with like huge magnets in their beaks, and they can still fly. So all those things right now, most of the results are consistent with the spin hypothesis. And for the cryptochromes in solution, there's no doubt that the only known way of explaining that is the chemical reactions that depend on spin. There are many chemical reactions that depend on spin, and for those, there is no alternative explanation on what's happening.
Right. Okay. Okay. Thanks for that. Yeah. So there's another question. Aritra is asking if we have any clues as to how quantum mechanical influences might influence quantum mechanical processes might influence neural activities at the cellular level.
So no, right? There are theories. There's no experimental evidence either against or in favor. There are many theories. We have just published a paper like disagreeing with someone's proposal on how that might happen. Here's what I think. Again, this is a hypothesis. We will be building an experiment to prove or refute this. This is not based on data. I have two hypotheses or two things that I think about. One is ion fluxes inside cells, right? Ion fluxes regulate stuff inside our cells, right? Fluxes of ions going in and out of the cells. I wonder if certain ions going into and out of the cell, those transient ion fluxes, if those could not be producing transient magnetic fields that alter spin-dependent chemical reactions. Okay, this is one hypothesis, not proven or refuted. Second hypothesis is the opposite. Like many of those spin-dependent chemical reactions happen attached to membranes. Like in birds, cryptochrome is attached to a membrane, right? For example, I wonder if spin-dependent chemical reaction process occurring in membranes, like cryptochrome attached to membranes, or lipid peroxidation. Membranes are lipids, right? Peroxidation is like a process that is spin-dependent, that is known to be spin-dependent, like in vitro. I wonder if spin-dependent chemical processes occurring at membranes might not be changing the functioning of ion channels, because ion channels are membrane channels, right? So those are the things that I think about, and there is no whatsoever proof to refute or prove that. Right?
Yeah, but these sound like very plausible things.
But there needs to be experiments, right? And that's the bottleneck right now with chess.
Yeah, I'm sure.
I'll tell you if it's what's that? Sorry.
I'm saying I'm sure it's very hard to recreate these experiments in the lab because it needs a lot of control.
Yeah, and that's why we need to start at like the single cell level using quantum techniques, right?
Yeah. So Matthew had he's put up a long comment here, but Matthew, if you would like to unmute yourself and discuss what you wrote in the chat. Or maybe Clarice, you can read it and
who is that?
Matthew wrote in the chat
Matthew.
a while back. Yeah, it's I think like 4 or 5 comments.
Are you open to the possibility of experiments ruling out life using spin physics? Oh, yes. And I think that if we can prove down to a certain noise of level that spin physics is not a thing, it still advances things, because then we need to come out for other ways why magnetic fields are affecting DNA repair, birds and stuff. And yes, that is a possibility, right? And I think that we can rule that out down to a certain noise level that we can. So totally, clearly, quantum knobs are there. I can imagine a scenario. I can also imagine a scenario where evolution did not dig deep to manipulate quantum effects. Yes, like, to come up with a molecule, maybe evolution. Yeah, sure. And yes, and right. For example, there are people thinking about, since you mentioned photosynthesis, there are some people thinking about the fact that photosynthesis might be quantum mechanics optimized, because like photosynthesis outperforms like human-made solar cells like by a lot, right? So one of the ways that people are thinking is that actually photosynthesis knows to how to do noise assisted transport in a quantum mechanical way. That is, it talks in a quantum mechanical way to noise in your system, to phonons in your system, to actually optimize how information is transferred, so that it makes this. So spins in biology is not the only thing that people are thinking about when they think about quantum knobs in biology. And I don't believe that we have a conclusive proof for any of those things. And I think there needs to be more high-tech experiments, right? And yeah, and I think there's still a lot to be done. And I think that we do need high-tech experiments as soon as possible, because otherwise it's always going to be like the chemists against the experimental, the behavioral biologists. So we need some quantumness in the game. We need. And if nature is exploring quantum mechanics, one reason why I like spins is because the lifetimes of spin processes is like much longer than lifetimes that have to do like ultra fast transfer and excital transfer. So ultra fast processes, like the ones that people think in conjunction with photosynthesis, they're like femtosecond timescales. Those are very, very fast. I like spins because spins give us some slack, because they happen between like nanosecond and microsecond. So I think that it might have been easier for nature to use those timescales instead. But again, not a proof, not just a hypothesis. More questions?
Okay. Yeah, I think Sarbajoy has a last question.
I like your picture.
Oh. Joy, we can't hear you. Oh, I think there might be a connectivity issue on his end. But okay, so I think we've all had a very nice time, and thank you so much once again. It was very interesting, very insightful, and definitely something that I'm sure all of us are going to go back and, want to read more about. I am pumped, really.
Thank you for having me.
This is very interesting.
So what I can, so I have one thing to recommend. It's a book called Life on the Edge.
Okay.
And I also have, you know what? I'll send you a link to the book. You didn't get it from me, but I'll send you a link to the book, like a PDF.
Okay.
I'll send it to you, and then you can to you both, and then you can like maybe share with people.
Yeah,
and I'll also send around, like, when people talk to me, like, so what are the articles on quantum bio that you would recommend? I have like a recommendation of those articles too.
Well, it sounds great. And yeah, I think everybody who's interested in quantum biology, there'll be tons of material for you to get started in, for you to, know what you're getting into and get started. And hopefully at some point, I guess there'll be a lot more people working in quantum biology, and maybe it won't take half a century. Maybe it'll be sooner because everybody will realize, oh my God, this could be the answer for things. And
Let's see.
yeah, and
Let's see
like
about that.
everybody is just. Yeah, no, of course, this talk has been very amazing. Thank you so much, Clarice, for the wonderful talk.
Thank you both for organizing. Thank you all for being here, and it's been a pleasure. I hope to continue to hear from you, and let me know if you want to talk.
I also
For
wanted to
sure.
thank you from like both of our sides. Thank you very much. It has been a very interesting talk, and I mean, the enthusiasm that you share about this field particularly is very contagious, and it is like making everyone excited about your work and the quantum biology field in general. So
Cool.
it
And
was
we're always
very
looking.
interesting and very sort of it particularly made at least me very interesting in this particular field, and I can see comments that are thanking you for this session, and they also seem very hyped up about this topic. Thank you very much. So would it be okay if they email you and their questions or anything they want to share
Yes.
about this talk?
Yes, my email is very easy. I put in the chat. It's just CLA, the first three letters of my name, at UCLA.edu.
Great. We will probably share it along with the recording also. We will be putting up
Yes.
this recording. Some people are asking about the recording as well. So we will be, I mean, I hope it is okay for you if we put up the
Do you
recording.
want the slides too? I can also share the slides.
Yeah, that
Yeah.
will be very helpful. Great. So
Thank you for having me.
Wow,
It was
No,
really
thank
there's
you.
nice.
someone in the chat who's like decided what their major is going to be after the talk. Also in ISO, we have like a 5-year program. The first 2 years, we study all the 4 subjects: physics, chemistry, math, and biology. And in the 3rd year, we
Cool.
choose our majors.
This is so cool. Can you do mixed majors? What about interdisciplinarity?
We can do majors and minors, but so as long as interdisciplinary goes, I've heard people in the physics department doing their projects under people in the biology department or the math department. I know somebody who was in the math major, but then he did a lot of research and his master thesis was on mathematical models in ecology. So yeah,
That's
people are
super
doing
cool.
people
Yeah,
are
there
doing
Yeah.
interdisciplinary stuff, and
Yeah.
I'm sure your talk has gone a long way in propagating that culture at ISO Mohali and the other ISOs.
That's cool. That's cool. I'm happy for that. If any of you want to talk, let's talk. And yeah. Thank
Yeah,
you.
wow.
Thanks for having me.
Well, thank you so much. It was our pleasure entirely.
Yes. It was my pleasure. Bye, people. Take care.
Yeah, so before we
Bye-bye.
leave, it has been a sort of tradition for our talks to take a group picture kind of thing. So to make
Yay.
it
Yes.
very near
Yes.
to offline experience. So if people are fine with it, then those who are still here, they can switch on their cameras and we can take a group photo.
Oh yeah.
Yeah, people are, this is so nice. I mean, this is the this has been a highlight of our talk since very
Cool.
first talk that we have arranged, and it makes it very personal experience. So it feels nice.
Yeah. Yeah.
Yeah. So okay. So say cheese, everyone.
Cheers.
Or say quantum, everyone.
Ooh, Cupid.
Thank you.
Great. Cool. Thank you very much. Thank you, Clarice, for in accepting our invite and for this very insightful and very enthusiastic talk. And thank you everyone for joining in and having such a wonderful interactive session. I will also thank Sonal and Shashwat, Smriti, Rimjim, the whole WiPMA and QTpi team. So yeah, on this note, we can probably end this session. Thank you again, Clarice.
Thank you all for
Bye.
being here. Thanks for organizing both